episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome, everybody, once again to another episode of our show, bringing you another fascinating guest today, helping to create a better tomorrow on so many different fronts. Very interesting topic today, as we're being joined by Dr. Leticia Toledo-Sherman, who is currently Senior Director of Drug Discovery of the Tau Consortium for the Rainwater Foundation, and she also holds an appointment as Adjunct Assistant Professor of Neurology at UCLA. Dr. Toledo-Sherman leads drug discovery activities for an international network of scientists that are working to develop therapies for Tauopathies, which is a group of neurodegenerative disorders characterized ultimately by the deposition of abnormal tau protein in the brain. Previously, she was the Director of Medicinal Chemistry and Computer-Aided Drug Design at the CHDI Foundation, which is a leading drug discovery organization developing programs for therapeutic use in Huntington's disease. At CHDI, she also led structural biology initiative that was critical to understanding the relationship between the structure and biological function of huntingtin, which is a protein that's mutated during Huntington's disease. Prior to CHDI, Dr. Toledo-Sherman was Executive Director of Chemistry at Lymphosign, which is now part of the Pharmascience organization that applied rational design principles to the development of therapeutics for blood cancer. From 2000 to 2004, she led a multi-site, multidisciplinary team using chemical proteomics and bioinformatics to discover various therapeutic targets and to investigate the mechanisms of actions of drugs. She started her career as a scientist at Kinetic Pharmaceuticals, now part of Amgen, where she implemented in silico platforms aimed at rational drug design of kinase inhibitors. And beside her work in the discovery and the development of therapies for neurodegenerative diseases, Dr. Toledo-Sherman is interested in the field of chemical biology. She has been involved in several initiatives and expert panels on the use of proof-of-concept chemical probes to validate targets and interrogate biological mechanisms. She's an advocate for open science at the pre-competitive drug discovery stage. Dr. Toledo-Sherman received her PhD in organic chemistry from the State University of New York at Stony Brook, did her postdoc work at MIT and the Skaggs Institute for Chemical Biology at Scripps Research Institute in La Jolla. Dr. Leticia Toledo-Sherman, welcome to our show today.
Well, thank you, Ira. Thank you for having me.
It's great to have you, and I would love to start off, as we typically do on the show, by handing you the floor for a couple minutes. If you can take us back a little bit early in the story, everything from where you grew up, when you first got interested in science and organic chemistry, and ultimately what led you into drug discovery and development. I think that's a great way to start things off.
That's going to go a while back, but that's fine. So I was born in Cuba, and I spent the first 15 years of my life in Cuba, almost 15 years of my life in Cuba. And while I was in Cuba, I was very interested in science from very early beginning, and I was encouraged, and I got involved in science as something called a monitor, or, you know, I was a kind of a TA in 7th grade, and I was keeping the glassware and the stockroom in order and kind of fantasizing about chemistry and the ideas that you could sort of make new things from, you know, existing things. So that came pretty early on. Our family immigrated to the United States around when I was 15 via Costa Rica first. And I continued thinking of science as really my primary goal. So it was always, I liked other things like art and literature, but really science was always my goal. And when I started my college career, again, it was, I was almost too determined to go into science. I wish that I would spend a little bit more time exploring other opportunities, but I became a chemist, and I had the fortune to be placed on a co-op program where a lot of the students graduating from the university that I graduated. It used to be Jersey City State College, or New Jersey City University now. had a co-op program from some grants that they got from the state of New Jersey for placing students in pharma and sort of industry, and I was fortunate to go to Ciba-Geigy.
Okay.
So at Ciba-Geigy, I got exposed to drug discovery and development, and I was mentored by a very amazing scientist, Dr. Frank Douglas, who was leading a lot of the pharma and, well, the New Jersey site in Summit, New Jersey at the time. But he also was encouraging students to actually go into the sciences. So I spent a total of 2 years at Ciba-Geigy, and I made up my mind at that point that I was going to go to graduate school and become a medicinal chemist. So I started, excuse me, I started my PhD career at Stony Brook, and originally determined to go into medicinal chemistry, but then because of funding issues, there was a material science project that was better funded with my the PI that I chose, Dr. Frank Fowler, in collaboration with Dr. Joseph Lauer. And we were, so my PhD project ended up being on crystal engineering. We were trying to learn from protein structures on how to orient how protein structures use hydrogen bonding. The amino acids in the proteins get assembled into three-dimensional structures. And they have these layer motifs, they have helical motifs. So we were learning on how to use those motifs in the hydrogen bonding, which are these forces that the amino acids use for the proteins to fold, to make layer materials for nonlinear optical applications. So you can imagine, nature cancels dipoles. So you have two molecules that have opposite dipoles. They usually go in opposite directions. So you get positive and negative canceling each other out. And we were trying to orient them so that they would keep the dipole, so they would orient into the same directions. And you need that to be able to have interesting properties in nonlinear optical, so you can change the frequency of the light going through them. So my PhD project ended up being one where we were, again, taking ideas from nature on how proteins and DNA are structured through hydrogen bonding and applying that to organic materials for these applications. But I developed tools of organic chemistry, obviously, because I was synthesizing these materials, crystallography, because I was having to understand how they fit together into a lattice, right? How they organize. And then computational chemistry, because that's how we were designing them. And it wasn't until I did my postdoc at MIT in a similar field called host gas chemistry or molecular chemistry that I really started to understand how all of that brought me back to medicinal chemistry, because those three tools or three disciplines really are the foundation of structure-based drug design, which is how medicinal chemistry uses protein structure and computational chemistry and organic synthesis to really create new drugs through the iterative process. So, you know, while I took this sort of detour in graduate school and my postdoc, it really was when I moved to Scripps. Our labs moved from MIT to Scripps, that I kind of saw everything happening around me in terms of structure-based drug design, and I sort of I would say re-educated myself on how to use these techniques for the purpose of structure-based drug design. So I started my career at Kinetics Pharmaceutical to work on kinase inhibitors. It was founded by Nick Lydon. He's a Lasker Prize winner. I was employee number one. I, you know, walked from my postdoc at Scripps in La Jolla to, a basement in Medford, Massachusetts, and learned how to do everything from getting the building, looking for sites, leasing all the equipments,
Yep.
designing the labs, doing all that fun thing, and ultimately building a team, and applied all of that technology that really I had very quickly put together, I think, or sort of like put that cohesive kind of package together in last year of my postdoc, to apply it to finding structure-based, or I would say structure-guided docking of compound libraries against protein structures of kinases to filter and enrich for bioactive kinase inhibitors, or compounds that were starting points for optimization of kinase inhibitors. And with a team of structural biologists and other computational chemists and medicinal chemists, led by Nick Lydon, we put together a very successful platform that led to the acquisition of the company by Amgen in the year 2000.
Sure.
And so then you've covered already where I've been since then, but that's kind of how I started my professional career, and how I got into doing structure-based drug design as a medicinal chemist.
Outstanding, really outstanding. And so you have this fascinating drug discovery story. But before we get to tau and tauopathies, I want to stop one other place. And I was reading an article, you were interviewed a little while ago, talking about your experience in Huntington's disease. And here again, you were talking about a terrible neurodegenerative disease, an orphan in many ways, that a lot of what is referred to, like many of these diseases nowadays, major unmet medical need. Talk for just a couple of minutes about your experience in the Huntington's disease space, and a little bit of what that taught you about some of these unmet medical neurologic diseases during your career.
Absolutely, and it's very related to what I'm doing here and the tauopathy space. So I joined the CHDI Foundation. I would say it's a nonprofit foundation funded with the sole purpose of discovering therapeutics for Huntington's disease. Now, Huntington disease is a monogenic disease.
Yep.
There's a 50, if someone inherits from parents that carry the genes, they have a 50-50 chance of inheriting a damaged copy of the gene, and that's all that it takes to cause the disease. So it only takes one of the alleles, or the bad copy of that gene, to actually inherit the disease. So the CHDI Foundation was formed by at that point it was an anonymous philanthropist to really speed the therapeutic development for Huntington's disease, which at that point and even now there was no therapeutics, or at least disease modifying therapeutics. So the Huntington protein is derived from the Huntington gene, and it has an expanded so normal the Huntington protein is about 3,400 kilodaltons. And it has an expanded polyglutamine repeat at the beginning on the end terminal, the beginning of this chain of amino acids. And that when the repeat gets longer than 36 of these glutamines, the protein misfolds. It's an intrinsically disordered proteins. These are proteins that are, they interact with they have lots of different function, and the disorder properties are ones that allow it to do lots of different functions because they interact differently. They present different structures to different interacting proteins in the cell. But that mobility, or ability to adopt so many conformations, it's one that it's through this expansion repeat it malfunctions and ends up misfolding, and it can no longer do the job that it's required to do, even though that function is still unknown, predominantly unknown. We try to understand it through structural biology techniques. But that protein misfolded ends up becoming aberrant in many of the processes that it's involved in, and therefore eventually cells dysfunction and die. So the idea is that we developed at CHDI through lots of different innovative techniques, from all the different omics and understanding all the interaction proteins that Huntington interacts with, and all the different cellular functions, how those functions are altered, was using all of these as entry points for therapeutic development. So CHDI developed a very large portfolio of therapeutic intervention points, small molecules, antisense oligonucleotides, lots of different modalities, drug modalities, to really get to correct that misfolded protein or the aberrant sort of mechanisms in the cell, by targeting the amount of protein that gets produced through antisense oligonucleotides to other modalities, as I mentioned. So anyway, there's a pretty robust pipeline of that CHDI has created and continues to work with in many pharma companies and biotechs and academics to continue to develop compounds for Huntington's. So I got recruited to come to the Tau Consortium, which is similarly concentrating on rare diseases. So the Tauopathies, or primary Tauopathies, are they're diseases that are caused or associated with similar misfolding mechanisms of the protein tau. In some cases, those mechanisms come directly from mutations on the 4R tau repeat of the protein that's involved in interactions of tau with the microtubules as they move in the axons. And those mutations can cause, in some cases, they're causative of disease, so some of the primary diseases. In some cases, there are more associated risks to develop disease. And like Huntington, tau is still poorly understood. And this is one of the biggest challenges with some of the rare diseases, is that, and especially brain diseases, is that the brain is complicated, many different cell types in the brain. So the function of a protein can change dramatically based on, the cell type, where it is, in which compartment of the cell it is, and also in, the time, the temporal time. Is it, is it developmental? Is it necessary for growth? Is it necessary for, adult onset? So the function of a protein can actually change as, with time.
Sure.
And so all of those things complicate the story of understanding the cellular function of tau, like Huntington.
Speaking about that, so you mentioned the primary function of tau, and I think in the public, probably the most common associations to tau, one, of course, the tau potential connections in Alzheimer's disease and dementia, and then I guess in sort of from the movies, the chronic traumatic encephalopathy from the concussion movie with Will Smith, another type of tauopathy. But then, in preparing to talk to you, I was going through sort of the grand list, and there's things on here that I've never heard of: meningoangiomatosis, subacute sclerosing panencephalitis, and then, of course, what started everything at the Rainwater Foundation, progressive supranuclear palsy. Could you sort of, I guess, putting Alzheimer's aside for a minute and the tau connection there, are the tauopathies, are they all orphans? How many are there? And then again, you're focused on progressive supranuclear palsy at the foundation, or that's sort of got this thing started there. But how do you decide? How do you decide which diseases to work on additionally to that, talk a little bit about that. And then sorry, a second, I know this is a multi-part question here. If you could take a couple minutes also and just talk about who Richard Rainwater was. I'm somewhat familiar with him from reading my financial magazines back in the 1980s, but a little bit of that history as well, I think, for people that are unfamiliar with the story.
Yeah, so maybe I think maybe it's okay if it's okay with you. I'll start with that part first, because then it really will lead into the tauopathy next. So, and I'm only almost 3 years into the foundation, so my history of Richard Rainwater it's somewhat limited. But he was an amazing human being, and his trustees follow his role, and having a very strong presence in philanthropy, both in education and medical research. So the foundation is in this 30th year. So it started mostly, very well-known financial person with amazing. I think he's considered the father of private equity in the United States, and but he used his foundation for funding education, especially K through 12 education in the Dallas-Fort Worth area. In 2009, he got diagnosed with one of the Tauopathies, PSP, and started a consortium called the Tau Consortium to find therapeutics for Tauopathies. Unfortunately, Richard, so he assembled an incredible group of neurologists and scientists to form this consortia and come together and share data and reagents and ideas, and really, break, the barriers of getting therapeutics to these rare diseases. And PSP was the primary one because that was the disease that he was diagnosed with. When he passed in 2015, I believe, I might have had that date wrong. He left the endowment to his family to continue as trustees, to continue the work both in education and medical research. And that has continued today in very, very strong ways, in funding a very large network of collaborators at different academic institutions across the US and some of UK, and now we have some PIs in other countries like Germany and Sweden, but continue really the work on breaking, understanding the disease and creating therapeutics for these tauopathies. So going back to tauopathies, there are about 20 different rare tauopathies. So when you put them all together, it becomes a fairly large number, and including some of the frontotemporal dementias that are caused again by frontotemporal dementias could be caused by other proteins, including TD43, but it's also caused by tau. So there's a subset of these dementias called Alzheimer's related dementia that are really caused or associated with the tau. The tau protein, as it is known in the Alzheimer's field, the neurofibrillary tangles are caused by, there's 6 isoforms of the disease. The adult brain produces 6 isoforms. The isoforms that they either have, they different in the domains at the end part of the protein, and also in the microtubule domain, which are these repeat domains that again bind the microtubule. So the 3R repeats, so proteins that have isoforms that have 3R repeats, so 3 repeats, so 3R meaning 3 repeats, are associated with the tangles in Alzheimer's and some of the other tauopathies. And the ones that have 4 repeats are usually associated with these primary tauopathies and some of the, encephalies. So there's that heterogeneity of the protein structure is actually another one of the difficulties in creating drugs for them, is because they are different and they interact with different proteins. They are in different locations in the cell. They have different functions. So that's actually one of the issues with, again, studying tau, is that a tau for an Alzheimer's disease, if it's causal in Alzheimer's disease, is a slightly different version of the protein that it's causal. So primary tauopathies are these non-Alzheimer's tauopathies, and then secondary tauopathies are related to this 3R tau and Alzheimer's. So that gives you a little bit of an idea of how complex this sort of landscape of protein structures and protein and different RNA sequences that translate into protein, and then again the temporal dimension of these, whether the early brain doesn't have much of this 4R tau repeat, but later in the adult brain it's expressed. So this gives you, again, if you're going to an adult disease, you need to have an understanding of cells that express that form of the protein as well. So it brings for some challenges in terms of the translation and drug discovery piece.
Sure. That being said, and you talked about your work in rational drug design, structure activity relationships, chemical proteomics, bioinformatics. As you undertake this research focus now, are you bringing other, we talk about tools on this show, all the new stuff coming down the pike from artificial intelligence, machine learning. What are you applying to sort of sift through this really combinatorial uniqueness of all these tauopathies? If you can just take us on a little bit of the journey through your current drug discovery process and some of the things that excite you about the new tools coming down the way.
Yeah. So I'll step back a little bit and tell you about the 3 areas of research of the foundation.
Please.
And then I'll depart from that to the drug discovery. But the foundation has identified, the Tau Consortium has identified 3 major areas of research. One is clinical enablement. So or clinical readiness. So that's funding observational studies and patient cohorts and defining rating scales and understanding the disease, right, from human population, and using samples from that human population to then understand the disease biology. So in this bucket of clinical readiness, we're again working with patient populations, getting them ready for clinical testing of interventional trials, but then really is to better understand the disease and better understand I would say the progression of the disease and what kind of outcome measures are going to be important, and biomarkers are going to be important for clinical trials. So starting from the human first, because I think a lot of the failures come from lack of understanding of what the ultimate human disease is. So that's a really kind of an important point, is that understanding from this clinical pathology to work back to understand the disease better by using different types of tools, really, to understand what are we learning from, let's say, from samples, from clinical samples from brains of diseased patients that have passed from pathology of tauopathies. What are they telling us about the disease? And from other types of measures, what do we know about translation? Then moving back into this basic discovery biology to really, understand better the disease at the cellular level, and understand which are the pathways and mechanisms that are really different between a healthy cell and a healthy brain and a diseased cell. So using all of the omics technologies that I'm sure you probably have covered in your show before, transcriptomics, genomics, metabolomics, protein-protein interaction, what goes wrong in these processes that are now different than in the healthy cell. And those are the mechanisms that are then become really good intervention points for drug discovery. So really defining what are the therapeutic mechanisms and targets, and creating models to be able to prosecute those mechanisms and targets through drug discovery. Many of the same individuals that have been studying those mechanisms have actually been hypothesizing how they can target those mechanisms to actually create drugs. So a lot of the portfolio of the drug discovery portfolio of the Tau Consortium really concentrates on taking these ideas or this hypothesis from this early discovery biology with the clinical cohorts already there, ready to really bring them through the so-called valley of death of drug discovery, which is taking programs from this academic setting, industrializing, bringing best practice drug discovery techniques to them. And we're doing that by placing them in a contract research organization and putting together a team of chemistry and biologists to then execute drug discovery the same way with all the tools, structure-based drug design or medicinal chemistry tools that about pharma or biotech would do, and really accelerate the development of therapeutics. But it's really in this space of early preclinical, early drug discovery to preclinical that the foundation concentrates. And we fund through RFAs of research, open requests for applications or proposals. We have a program with the Alzheimer's Association where we're capturing a lot of those new therapeutics mechanisms and ideas outside of the Tau Consortium. We have another effort both at the Tau Consortium and outside of that through a collaboration with the Michael J. Fox Foundation called Pipette, which is to develop PET agents as tools for diagnostics and clinical trial therapeutic testing. So those are all sort of translational programs, but we're all applying this similar set of best practice drug discovery tools to advance those programs towards the clinic. And that's really my main role at the foundation. It's help taking these programs from this academic setting where a professor has these amazing ideas and lots of different ideas, and figure out which are those ideas that are best, where applying rigorous target validation ideas and identifying hits through virtual screening or medicinal chemistry, and then take them forward. And we have a portfolio right now of about, I think, 30 something different programs by the time that we put all of the Tau Consortium and the TPET programs with the Alzheimer's Association and Pipette together. And so a lot of lots of diverse mechanisms where we have programs that aim at lowering the amount of tau that gets produced by stopping the synthesis of the target protein. We have programs that aim by different mechanisms to correct that misfolding, that proteostasis machinery. So the cells have a lot of the whole cellular proteostasis, which is this homeostasis state where all the proteins are folded and refolded properly in a functional manner. There is mutations or these, other effects on the cells. The misfolding can go awry. identify where to intervene, then we can correct that misfolding back to sort of normalcy. So the idea of this proteostasis is that we push back the landscape of folding and refolding to the normal state. And that's by you can deplete or decrease the amount of misfolded protein. You could enhance it by working with chaperones. We can degrade it by the proteasome or autophagy. So lots of different sub-mechanisms by which you can do that correction.
It's really fascinating. This is a this is a side question. I apologize if this is if this is stupid, but I was just wondering off the top of my head. You know, obviously we have the beta amyloid component of Alzheimer's disease, and now we're learning all about these systemic amyloidoses.
Yeah.
Are there tauopathies of other parts of the nervous system, or things that look like tauopathies in other parts of our bodies? Is there some?
Well, there are other diseases that affect similar. They're different amyloid proteins. They're not tau.
Right.
Tau, perhaps tau is associated, but there are other proteinopathies that are. So, for example, tafamidis is a new drug that is to correct a proteomyopathy of the heart by this transretinine protein. And one of the PIs that we fund, Jeff Kelly, is the developer of tafamidis as a drug.
Okay.
And he's working towards doing the same thing for tau that he did for the transretinine protein.
Interesting.
But you know, I think tau has mostly been studied in the brain.
Yeah.
But and it's mostly expressed in neurons, but there are other cell populations that are studied. One of the really fascinating things is that the tangles have now been associated with non-disease states. So, for example, hibernation is one where the tau exists in a tangle state in hibernating animals. So we know that the tangle itself is not always associated with disease. There are conditions in which the tangles are associated with some other state, and that function is still really not clearly elucidated yet. But we know it's not always, tangles and disease are not always one-to-one. So that's a very intriguing observation.
It's interesting because I just along those lines, I had a guest on a little while ago talking about hibernation. We were talking about Arctic ground squirrels, and she was mentioning how we don't think about it, but during hibernation, the CNS, it falls apart. A lot of it, they wonder, wow, how does this little squirrel fall asleep, and then they pop back up many months later and everything's nice and clear. But even though a lot has been destroyed, and it's just interesting because you're saying at the beginning this tau does important things for us, and it's just that's a really interesting finding. I hadn't thought of that interview for a while, but that's a really interesting thing.
Fauna Bio, was this?
What's that?
Was your guest from Fauna Bio?
No. I know Fauna. This was Dr. Kelly Drew up at the Arctic Biology Center at the University of Alaska. So she studies the ground squirrels up there. But I'm familiar with Fauna.
Yeah, because they're trying to develop some tau models that are again more based on the hibernation states and understanding that biology.
Really cool. Really cool. Leticia, some next steps. Any obviously you have a major portfolio, a lot of things to work on. What can we expect in terms of 2022? Are you going to be at a lot of conferences presenting? Where we can follow you and watch your continued success in driving this all forward?
So there's going to be a big conference. Well, at the later this month, there's going to be a Tau conference. It's called EuroTau. It's a conference that is sponsored. It's going to happen in Lille, France.
Okay.
I will not be there, but colleagues from the Rainwater Foundation will be there, and they will be delivering the the Rainwater Prize program, which is a prize program, again rewarding Tau research. And the winners of the 2021 prize will be awarded at the conference. Next year, there will be a Tau conference in Washington, D.C. around February called Open Tau 2022. It's the second year in a row that there will be a Tau conference worldwide, with partnerships from the Cure PSP, Alzheimer's Association, and the Rainwater Foundation. It's going to be a very interesting agenda with lots of interesting programs and some of the therapeutics that are happening. So the Tau Consortium it's funding Tau therapeutics, but many pharma have their own programs.
Okay.
And that becomes this Tau, Open Tau is a venue by which there's reporting from both pharma and research foundations and academia. So that's a really amazing forum in Washington, D.C. next year.
Got it.
So we have a lot of initiatives. We're publishing some of them. Some of them are taking a little longer. The most important thing that we're doing is we're really partnering with drug discovery institutes across the programs that we support, so that we can bring expand on the ways that we can do integrated drug discovery. So we've done a couple of programs with a CRO, and one example which was really successful with was a program from Matt Disney on small molecule modulators of tau protein. And we took that program and we took it from his lab, and so the model is to continue to have the PI involved,
Okay.
then assign a drug discovery team, and then eventually move that program to a place so they can be commercialized. So that model was executed over COVID. Actually, we took that program into Charles River. It's one of the major CROs, and they did integrated drug discovery and structural biology, and Matt Disney continued to do NMR and assay development, et cetera. And then that program has now been in-licensed by Expansion Therapeutics.
Okay.
Yesterday announced that they were able to raise $80 million in funding for neurodegenerative diseases, including Tau is not their only program. They have programs in ALS and other programs, but that was a major sort of a component of the assets that were used to raise money. But it was our proof of concept that we can actually do this. Take a program from an academic setting, industrialize it at a CRO or a drug discovery institutes, and move it towards a place that they can then be a standalone biotech that can raise money or partner with pharma, et cetera. And so the new version, or the next version of these, is actually we're partnering with the Drug Discovery Institutes of Alzheimer's. They're funded from Alzheimer's Research UK, Alberta, Oxford, and UCL. There are 3 institutes, and we're coming up with three-way. We're negotiating currently on these 3-way collaborations. So expanding the Tau Consortium really to then have these collaborative enablement of drug discovery to be able to, again, move these programs from this academic setting to more of an industrial setting. So that's kind of the next iteration. So we're spending a lot of efforts to coming up with a refinement of that model. So we expect that once that has been implemented, we'll be able to start kind of moving a lot more of these programs forward.
Very exciting. It's, it's very refreshing, as you were saying. It's not just to see, the unmet medical need combined with getting across the valley of death, and now collaborative models. We don't normally see pharma collaborating as much as, but now, we're getting to realize, yeah.
There are a few examples of that successful collaborator at the pre-competitive stage, and one of them is the Structural Genomics Consortium. I don't know if you've had anyone from the genomics invited, but it's very refreshing, because if you can sort of bring the platform higher, instead of everyone sort of reinventing the wheel at the pre-competitive stage, you can bring pharma together and develop tools to a certain point, then that could be very higher level departing points, and each pharma can go in and create their own sort of more refined drugs behind their pharma walls, but starting and sharing a lot of this pre-competitive enablement before that. And so Structural Genomics Consortium is a good example. We're bringing open science and a lot of that enablement to the Tau Consortium, and then again, this kind of Uber consortia of now working with the Alzheimer's Research Institutes to do that is another way that we're trying to kind of remove a lot of the barriers and redundancies of science that happens in medical research.
Yeah. Very, very fascinating. And it's an amazing path you're on here, Leticia. I really wish you the best with all of this and leading it forward. For everybody that's going to be listening to this particular episode on our podcast or watching on the YouTube channel, you've been spending time today listening to Dr. Leticia Toledo-Sherman, Senior Director of Drug Discovery of the Tau Consortium for the Rainwater Foundation, also Adjunct Assistant Professor of Neurology at UCLA. Leticia, I want to take time to thank you for coming on the show to talk about everything you're doing and all your visions. Thanks for doing it. And as we say on our show here, thanks for helping to create a better tomorrow through all your hard work. Very, very inspiring story.
Thank you, Ira. It was a pleasure being here.